No.
Yeah. Okay. I can start. If it's okay, it will be. If it's okay, it will be in English again. I'm sorry. I also now went back full time in the lab, so my Swedish is disappearing again. So I'm trying my best. But yeah, I will start. I'm just gonna tell you just my story, how I ended up doing a PhD and what does it mean to start with. And I would say this is a nice representation of me in the lab. That's not me, but this is what we are usually doing. We are sitting there and we're pipetting teeny tiny things over and over and over and over again. So I'm going to talk about my adventure that I started, but a bit about myself. So my dad is from Syria, my mom is from Russia. I grew up in Jordan. So when people ask me where are you from, it's always a complicated question. I can never say I am from Jordan, but then they start asking things, and I'm like, well, yeah, I'm not actually from there. So it's a mix. But I grew up in a mixed culture because my mom was Russian, my dad was Arab, and I grew up in an Arabic country. So there was a lot of interesting things. It was a bit different for me than it was for the other people because I always had other things that, yeah, the people didn't understand, or celebrations that we did and they didn't have, and etc. But I started here. I did my high school or the gymnasium with the Naturvetenskap track. We have a similar thing. And I finished my high school with really good grades, and there was the question, what do you want to do next? When I was growing up, I was obsessed with detective series. I loved them. I loved Sherlock Holmes. I loved CSI and CIS, etc. And for me, the most interesting people in them were these people in the forensics labs. The people who are taking the fingerprints, the ones that are taking that hair that they have found and going and checking for the DNA. So I told my parents, I want to be a forensic scientist. And they told me, no way. They were like, nope, not gonna happen. It's not a job that, good for those countries for several reasons, mainly security. And you're a woman, so it was a no-no. Then I had several interesting suggestions. I wanted to be a mortician, the person who works with dead bodies. They were definitely not with that. And a couple of other ones. But I really knew that I loved biology, I loved chemistry, I loved science. I didn't like physics whatsoever. So I wanted to go in that direction. So in the end, we ended up with finding something in the middle that was acceptable and good in the culture and etc. And I went and I did a Bachelor of Pharmacy. So pharmacology. That was 5 years. We learned everything about the drugs. From the teeny tiniest molecules, how do you design them, make them a tablet? How do you give the tablet to the patient? Which patient do you give them to? What happens after the tablet is in your body? Where does it go? How does it dissolve? Which cell does it attack? And so on. So quite a lot, a really, really broad spectrum from like the tiniest chemical stuff to actually dealing with patients. This is us on our last day where we were throwing our lab coats that we were super proud of to show for everyone that now we are official pharmacists. Yay, we are done. What is next? So I knew I didn't like being in a community pharmacy, so like in the apoteket, it wasn't for me. So, and I really, really wanted to do research. So there is no such thing as a lot of research in Jordan. It's a very small and slightly poor country, so there was not a lot of research, and I decided to move next. So I found a scholarship, and I went to Netherlands, said bye-bye to my parents. They were shocked. There was a lot of judgment from the society because I was a girl and I was going to travel alone, and how am I going to live alone in a foreign country, and etc. and etc. But yeah, meanwhile I moved to Netherlands and I started my master's degree. So I did Biomedicine and I specialized in toxicology. And that is basically where you're looking at the adverse reactions of the medicine. So every medicine that you take might have some side effects that you don't want, but it also, we are studying chemicals that you are having in your everyday life, the disinfectant that you are using, the water that you are drinking, and all of these things, whether it's good or not. And that is an example of a test that we do to check whether a chemical can cause cancer or not. and a lot of reports and homeworks and so on. So that was me. It was a big change for me. We were not used to the way it was, the study that was done. I never had like this self-study and group work and all of these stuff. So that was a shock. And I did 2 internships. So this practique. The first one, if you read this, it sounds like a very fancy, complicated title. But basically, I worked on kidneys, and there is a drug that is called cisplatin that they give for people with cancer. And that drug is known to cause really bad side effects on the kidneys. And we were studying why. And we found out that there is one transporter that sits on a cell, and the presence or absence of it causes a huge difference. So if it's there, you don't have a lot of toxicity. If it's not there, there is a lot of toxicity. Then another practice that I did was now in a pharmaceutical company. So it was like in the industry. And there we worked with something called antibody-drug conjugates. So the antibodies, now you have heard a lot about them in the news, the anti-crop, the things with all going on with the COVID. And what they did there is that they attach a small drug molecule to that antibody. And that antibody knows exactly which cell to go to. So this is a cancer cell, so that antibody will specifically go to the cancer cell and kill it. But it will not go to any other cells because it is only designed against that cell. So that was, again, also really interesting. Then moved on, decided, okay, I want to do the next step. And this is the PhD part. I never stopped studying. I keep on going and going and going. So then I moved to the next part, which is here in Stockholm. And I went to Karolinska. So this is me in front of Karolinska Institute. Yay, super happy that I made it to Sweden. By the way, I really hate the cold. And all my friends are keep making fun of me that I keep moving north and then complaining non-stop that it's too cold in Sweden. But yes. So that is the lab. Me on a normal day in the lab, you can see. And then this is the office with a lot of sticky notes and stuff. And the people you work with a really lovely t-shirt that says, I love research. Because we do. And what we are doing now is that we are breaking down the kidneys. I'm still working on kidneys, so we are breaking them down to teeny tiniest molecules. So this is our human body. We have a lot of different systems, organ systems. I am working on the urinary tract system, and I'm really focusing on the kidney part. So the kidney is made also out of different parts, and this is called the nephron. So this is the part that filters out your blood and produces the urine and cleans the blood from the toxins. And it is made, we keep on going deeper. I'm still like only halfway. So we go into this part, which is called the glomeruli, and this is where the filtration happens. And we go inside, and you can see there's a lot of different cell types in here. And each of these cell types do a different thing. So this cell is not similar to this one at all. And each of them have a really completely different function and a different structure and a lot of different things that are happening. So this is where the filtration is happening, and we are focusing on these 3 cell types that we like, called podocytes, mesangial, and endothelial cells. A lot of fancy names, but it's just like cells that are contributing to the filtration. And this is when we move in, and this is inside the actual cell. Yeah, and inside the cell you find the genetic material. And this is the DNA, and that is what I think you have known, you know this from your biology, that this is the DNA and this is the RNA. So this is the genetic code, and this is what we are studying. So what we are doing is that we are taking all of this in consideration, but we are looking at this part. So we check this, and then we try to see how it affects all of that up until the body. And we are doing it, of course, in the kidney. So in the we check how it is in the normal situation when you are healthy, and then when there is danger, so when your kidney is sick. And then we compare them. So we check how is it in the normal, how is it in the sick, what has changed in that code, and we try to figure out how to solve it. I can go into a lot of details about my research, but this is the disease that we are studying. It's called chronic kidney disease. It's a bunch of different diseases, but it means that your kidneys are not functioning anymore. It affects 1 in 10 people in Western countries, so it's quite a big number. That means, yeah, 1 person in this room. Like, this is the number when you think about it. It's 1 in 10. And it's a very, very costly disease because you need to either have a whole new kidney or you need to have like a dialysis, a cleaning done in the hospital several times a week. If you have diabetes, if you have hypertension, if you are smoking, if you're obese, all of these can contribute to it. So there are high numbers. And the problem is you cannot fix it. We can only slow it down. You can never make the kidney perfect again. So what we are trying to do is to understand what is happening in the disease, to hopefully be able to treat it, hopefully be able to switch it back, or to slow it a bit more. And my fun part, what I do in my daily life, is that I study a lot of mice. We have a lot of mice that we are working with. We modify them genetically, so we play with the different codes and see whether it makes the disease better or worse. We have human samples that we get from people who get their kidneys removed because they are not working anymore. We take pieces of that. Very nice, yeah, sorry. And we do a lot of cell culture. So we have cells that are isolated and we grow them and we test things on them and then we check them. Yeah, so when the kidney is functioning normally, if you do like an analysis that is called the GFR, you will get something between 60 and 120, which is great. And if it goes below 60, you can have a disease, and below 15, your kidneys stop working. I'm just going to show you a couple of examples. And why is that? Because then this is the filtration barrier we talked about. And when it's fine and everything is great, your urine only has urine. It doesn't have any proteins in it. But when it's leaking, because it's not working, you will get protein in it. And this protein we can detect. So this is an example from an actual experiment that we have done. Sorry for these details, but we took urine from mice and we run them. And you can see this is a healthy one. You see no protein. However, when they are sick, you see a protein here. So we know that this mouse is sick, for example. I'm just showing you like really small examples of the things that we're actually working with. This is a section, so we can take a piece of the kidney and we color it with special reagents and chemicals, and we check the structure. So you can see here, this is the glomeruli that we have talked about, the filtration, and these are the tubular part. They all look nice, and this is when it's sick. You see everything is mashed, crumbled together, and you can already see it under the microscope that it's not in a great shape. This is really bad kidney. Like you can see it. Some of them you can see like in the middle. Some of them are okay, some of them are not, but this is really bad. And we can go even inside. So these are actually inside the cells. This is inside this one cell, for example. You can see this. And you can see, for example, here everything is intact in one piece, and here you see a lot of things going on. So yeah, I can go on and on and on and on about my research, but this is just kind of like a very short recap on what you can be doing if you're working as a researcher. And there is a lot of different researches happening. We have groups next to us who are working with cancer, some of them that are working with brain, with heart, with muscles, with everything that you can imagine. Also like some, let's say, I don't know, psychology-based, mental health, yeah, and surgery, things with the patients, and so on. So if you're interested in it, maybe this is something you can think about. Yeah, and I think in the end, like one thing that I wanted to mention is that since I moved quite a lot from different countries, there was a lot of culture shocks for me, like especially at the beginning when I first came to Europe, and I was like, oh my God, like what is this? Like this is so different from what I'm used to. And also due to my work, I work with a lot of international environment with a lot of different people who have different cultures, traditions, languages, the way that they do things. So it's very important to be flexible and keep it in mind that not everyone thinks the same way that you do, not everyone was raised the same way that you have been, and just like be really flexible with it. So sometimes you need to change, you don't need to lose your core, let's say, or your true self, but you need to adapt some of your let's say actions or something like that. So yeah, this was in the end. Next step, I don't know. I hope that I will be finishing this year, and then we will see. It will be to be continued. I don't know where we will end up, but yeah, that was it for today.